Okay, so in this video, we're going to look at the genetic engineering technique of plasmid-based transformation and talk about some reasons why it would be used. So in plasmid-based transformation, um, in order to understand this topic, I would watch the previous video called Introduction to Biotech and Restriction Enzymes. So here we have a population of bacteria, and if we are doing a lab where we have some like plasmids, and hopefully some of them are recombinant plasmids. There may be some plasmids that are not recombinant, but we expose some bacteria to recombinant plasmids, and we hope for transformation, which transformation is when you take DNA up, or not you, but a bacterium takes DNA up from its environment. So when bacteria do this, as scientists, it's going to be kind of difficult to try and figure out which of these bacteria were successful at actually taking up the plasmid, as well as which one has recombinant plasmid, because it's not going to be 100% successful. So, and not every plasmid will be recombinant. So how do scientists identify which bacteria had successful transformation and a recombinant plasmid, right? Oh, sorry, this is for my class. So when we look at, here we go. So to refresh our memory, doesn't that transform bacteria look like it's a scared ghost? But here, when we talk about a transformed bacterium, that is a bacterium that has taken up the plasmid. So that dark circle that you see here is actually the main circular chromosome, and these two eye-looking things are the recombinant plasmids. So if a bacterium, though, takes up a recombinant plasmid, when that bacterium undergoes binary fission, it's going to make copies of that plasmid. So over time, you're going to have a whole group of scared bacteria, or you'll have a whole group of transformed, or colony of transformed bacteria. So if you are a scientist, though, and you're looking at a petri dish, how would you know which ones have those recombinant plasmids, right? So when we think about, like, there you go. When we think about this, here is one of the ways. When scientists introduce or use plasmids, they can actually use a genetically engineered plasmid that has a region on it that carries the gene for antibiotic resistance. So antibiotic resistance means that the bacteria will survive and reproduce even in the presence of antibiotics. And so some bacteria have a mutation on their plasmid that confers or gives resistance to antibiotics. So what that means is when scientists are working with plasmids, they genetically engineer them to already have that gene of antibiotic resistance to start. So to summarize so far, scientists can make agar. So it's not really just pouring antibiotics on top, but when the agar is liquid, they can like drop in antibiotics. And so as that agar becomes like that gel, like jello texture, there'll already be antibiotics spread within that agar, that nutrient agar. So the petri dish or the slant will already be made with antibiotics embedded. So if you have two bacteria and one of them has a plasmid that has antibiotic resistance gene and one doesn't, when you place these bacteria or when you grow these bacteria on the petri dish, you want to think to yourself, well, which ones are going to survive in the presence of antibiotics? It's going to be the one that has the gene for antibiotic resistance. So those will be the only, oh, I like that. So the ones without antibiotic resistance will die, and therefore the ones with antibiotic resistance will survive. So this is kind of like natural selection, survival of the fittest. Only the ones with a certain phenotype are able to survive and reproduce in that environment. So when scientists do transformation, it's not always certain that every bacterium will take up a plasmid. So then later, when you expose them to antibiotics, only the ones that had successful transformation will survive. That is super important to understand, that if transformation did not occur, that means they do not have the gene for antibiotic resistance, they do not have the plasmid, and therefore you know that all of the survivors, all of the colonies growing, contained the gene for antibiotic resistance, and where they got it was your plasmid. Okay, so again, let me summarize because it's like this is key to understanding. So if you start with a petri dish with antibiotics in the nutrient medium and you plate it, so you streak some bacteria across that petri dish. Now, only bacteria that had successful transformation and have the gene for antibiotic resistance will survive and reproduce. How did they get that gene for antibiotic resistance, right? They got it on the plasmid. Now, here's the other important part. What might else be on that plasmid? Well, hopefully it's the gene of interest. Whatever gene the scientists are hoping to make copies of or to express is also on that plasmid. So the gene for antibiotic resistance is used to help scientists identify which bacterial colonies had successful transformation and could possibly have our gene of interest. So if I look here at these two petri dishes and I want to think like, well, which one had successful, which one is actually like being grown on antibiotics? I would say like B. There's fewer there. That probably means that the ones didn't survive who didn't have the plasmid. Okay, so true or false, all of these bacteria are the result of successful transformation, assuming that that petri dish has antibiotics. True. Okay. So let's go ahead and you can pause here and try this question if you want. Now, when we talk about plasmids and having the gene of interest, so scientists will start with a plasmid. And when we look at this plasmid, you see a couple different areas identified. You have the promoter region. And if you remember, a promoter is where transcription begins. That's where it signals the start of the gene. And then you have your gene of interest that you're working with. Now, hopefully, let's go back to that previous video on the intro to biotech. So you start with a plasmid, and the scientists will start with a plasmid that has the gene for antibiotic resistance already, which in this picture is dark blue. And then they will use restriction enzymes, and they will cut DNA from another source as well as cut that plasmid to produce sticky ends. And when they produce those sticky ends, now hopefully your gene of interest will form recombinant DNA, which here is the green. So this would be a recombinant plasmid. The green area represents a gene from another organism. The blue area is the gene for antibiotic resistance, which helps scientists identify which bacteria were successful at transformation. And the orange that says ORI, that is the origin of replication. That is where DNA helicase will attach to make copies and replicate this plasmid. Okay, so when we look at another picture here, we can see a couple things. So in another plasmid, you have the AMP-R. So when they talk about a gene for antibiotic resistance, so this right here, ampicillin, is an antibiotic. So if a bacterium had this plasmid, it would be resistant to the antibiotic ampicillin because it has the gene ampicillin for ampicillin resistance. Now, the LAC-Z gene is a gene for bacteria that help it to digest lactose. And when you notice, you can look at these little lines and tick marks here. So the blue arrow says, what are these? Those are restriction sites. That is where restriction enzymes are going to cut. So EcoWAR1 is the restriction enzyme we talked about in that previous video. So if I expose this plasmid to EcoWAR1, it's going to cut it in that LAC-Z gene. So let's think about this for a minute. Let's think about a couple thought-provoking questions. So what would happen if the gene of interest was inserted into the LAC operon? So if right here in EcoWAR1, the restriction enzyme cut the LAC-Z gene, could this bacteria digest lactose? And the answer is no, because the LAC-Z gene was disrupted and it won't make the enzyme, because now you've literally cut in the middle of a gene and put human DNA in there. and mix it up. So now the bacteria won't be able to digest lactose. You just disrupted its region that codes for lactose. Now, part B. What would happen if the gene of interest was inserted into the AMP-R region? Well, that gray region is where the gene for antibiotic resistance is coded for. So if you cut in the ampicillin resistance region, you're going to disrupt that gene, and now the bacteria will no longer have resistance to antibiotics. Now, what would happen if the gene was inserted into the origin of replication area? Well, maybe it wouldn't be able to replicate. Okay, so it's important to remember that the plasmid is a sequence of DNA that does code for genes. So wherever you cut that plasmid, you may inadvertently be cutting in a gene, and now that protein product will not work. Okay. So here is a question. I believe this is from like the 2013 AP biology test you can find on the internet. So you can check yourself kind of on understanding this. And yeah. All right. Good job. We'll stop there.